This is kind of a hefty section of information, so hang in there. This is probably going to be about 20 minutes long. Selective breeding was really only able to be done based on phenotypes up until recently, the physical characteristics that breeders could see, because they didn't know how to manipulate the genotypes, the genetic characteristics. But what can happen now is genetic engineers can transfer certain genes between organisms to meet specific needs. It's easy to extract DNA from cells and tissues, and it can be cut into fragments of manageable size using what we call restriction enzymes. These restriction fragments can be separated according to size using electrophoresis or another technique. Here's a diagram of what happens with this. You have the recognition sequences, and as we had discussed, each restriction enzyme has a different recognition sequence. So you have those. In the recognition sequence, a cut is made, which generates sticky ends. The DNA is loaded onto a gel. An electric current is run through that gel. And then, just to refresh your memory, the longer fragments don't travel as far as the shorter fragments. That creates a DNA fingerprint. In 1987, Douglas Prasher wanted to look at the jellyfish genome to figure out what made jellyfish glow. He called that protein the GFP, green fluorescent protein. It absorbs energy from light in order to cause the jellyfish to glow. He thought that GFP from the jellyfish could be linked to a protein when it was being made in a cell, like attaching a light bulb to that molecule. To find it, he compared part of the amino acid sequence of GFP to a genetic code table and was able to predict a probable mRNA-based sequence. So he used that translator that you guys learned about last chapter and came up with the probable mRNA-based sequence that would code for this sequence of amino acids. He used a complementary-based sequence to attract an mRNA and would bind to that sequence by base pairing. Then he screened the genetic library of thousands of different mRNA sequences. He found one that bound perfectly, which was the mRNA transcript of GFP. So then, to find the actual gene, he took a gel in which restriction fragments from the jellyfish genome had been separated and found that one of those had bound tightly to the mRNA. That was the gene for GFP. This method is called Southern blotting, after its inventor, Edwin Southern. So here's what Southern blotting looks like. After that gel electrophoresis has been performed, you have the DNA banding pattern here. Literally, the bands on the gel are blotted up onto paper, and then this is right here, this is paper towels on top. Then DNA can't be visualized. It's kind of clear. So it needs to be made radioactive. The radioactive probes bind to fragments with complementary base sequences, and then there's an X-ray that's taken of it, which is called an autorad, or autoradiograph. Today this is computerized. It's quicker and less expensive that way. Once biologists find a gene, a technique known as PCR allows them to make many copies of it. So here are the steps to PCR. First, DNA is heated. Anything that heats expands, and in this case the DNA separates into two strands. At each end of that original piece of DNA, a biologist adds a short piece of DNA that complements a portion of the sequence. This is called a primer. They're called primers because they prepare or prime a place for DNA polymerase to start working. DNA polymerase copies the region between the primers, and these copies serve as templates to make more copies. So, and this is an exponential multiplication of the amount of DNA in a sample. Just a few dozen cycles of replication can produce billions of copies of the DNA between the primers. Scientists started wondering if DNA could be changed in a living cell, and they kind of looked back on Griffith's bacterial transformation experiments and thought, this has already been done. Remember that during transformation, a cell takes in DNA from outside the cell, and that DNA becomes a part of the cell's own genome. In Griffith's research, remember, there were heat-killed bacteria whose DNA was taken up by live bacteria. That's transformed the live bacteria and caused it to become harmful pneumonia-causing bacteria. Scientists can produce custom-built DNA molecules in the lab and insert those into living cells. Machines called DNA synthesizers are used to produce short pieces of DNA, and these synthetic sequences can be joined to natural sequences using DNA ligase that links everything together. In this way, a gene from one organism can be attached to DNA of another organism. Restriction enzymes cut DNA at specific sequences called sticky ends, just like we've seen before. So the same restriction enzyme is used for two different types of DNA here. We have the blue DNA from one organism and the red DNA from another organism. So now these sticky ends will match together. So now we've joined together DNA from the blue organism and DNA from the red organism together. This is called recombinant DNA. We have recombined the DNA. Now it's from two separate organisms. Recombinant DNA technology changes the genetic composition of living organisms. By doing this, scientists can look at the structure and functions of genes. Scientists who were researching recombinant DNA found out that many of the DNA molecules they inserted just vanished because the cells didn't copy the added DNA. It wasn't recognized as needing to be copied. It was foreign DNA. So what they started doing is joining recombinant DNA to another piece of DNA containing a replication start signal. This way, whenever the cell copies its own DNA, it copies the recombinant as well. Bacteria not only have a single chromosome, but sometimes they contain small circular DNA molecules known as plasmids. What scientists have done is joined this recombinant DNA to the plasmid and then use that to transform the bacteria. Results in the replication of the newly added DNA along with the rest of the cell's genome. So this is a plasmid from EcoR1. I'm going to walk you through this. This ORI right here is the origin of replication. That's the genetic start signal for replication. There's also a restriction enzyme cutting site here, EcoR1. Restriction enzyme cutting site. So that's a recognition sequence or a restriction sequence. Plasmids are also found in yeasts. Yeasts are single-celled eukaryotes that can be transformed with recombinant DNA as well. Biologists working with yeast can construct artificial chromosomes containing centromeres, telomeres, and replication start sites. These artificial chromosomes simplify the process of introducing recombinant DNA into the yeast genome. Bacteria can be transformed using these recombinant plasmids. Scientists can insert a piece of DNA into a plasmid if both the plasmid and the target DNA have the same sticky ends, as we've already discussed. So here we see recombinant DNA being made. Here's the gene for the human growth hormone that's been cut out of human DNA. We have the same EcoR1 restriction sites here. This is a plasmid from a bacterial cell with the EcoR1 restriction site as well. Add in the EcoR1 restriction enzyme. It all matches up. DNA recombination here. Now we have a bacterial cell containing the gene for human growth hormone. Same thing's been done with human insulin. Synthetic or lab-produced insulin is done using bacterial cells. So now that new plasmid, that new combination of genes, is returned to a bacterial cell, replicates the recombinant DNA over and over again, and produces human growth hormone. The recombinant plasmid has a marker such as a gene for antibiotic resistance, and this is because it makes it possible to distinguish bacteria that have been transformed, that carry the plasmid, from those that do not. This plasmid has two genes for antibiotic resistance, to tetracycline and to ampicillin. So after transformation, the bacterial culture is treated with an antibiotic. Only those that have these resistance genes will survive. So they'll grow this bacteria on a plate that contains tetracycline and ampicillin. So only the bacterial cells who have resistance to those, in other words, have contained this plasmid, will be able to grow. Organisms that have genes from another species are called transgenic. So that bacterial cell that was just demonstrated to have been created is called transgenic bacteria. They're produced by the insertion of recombinant DNA into the genome of a host. The DNA molecules used for transformation of plant and animal cells contain genetic markers to track that transformation. Transgenic technology was first used on mice in the 1980s. Now transgenic organisms include plants, animals, and microorganisms. By examining the traits of a genetically modified organism, you can learn about the function of the transferred gene. Plant cells can be transformed using agrobacterium. In nature, this bacterium inserts a small DNA plasmid that produces tumors in a plant's cells. Doesn't sound like a good idea to transform cells like that. What scientists have done, though, is they deactivate the plasmid's tumor-producing gene and replace it with a piece of recombinant DNA that can be used to infect and transform plant cells, and the transformed cells can be cultured to produce adult plants. So here we have the gene to be transferred. The transformed bacteria introduce plasmids into plant cells. Agrobacterium inserts part of its DNA into the host cell chromosome. A complete plant is generated from the transformed cell. So first we have transformation of the bacteria here, and then the agrobacterium, that bacterial cell, inserts part of its DNA into the host cell chromosome. And then a complete plant is generated from that transformed cell. There are other ways to produce transgenic plants also. If we get rid of their cell walls, sometimes plant cells will just undergo transformation on their own. DNA could be injected into cells. If transformation is successful, the recombinant DNA is integrated into one of the plant cell's chromosomes. With animal cells, some of the same techniques are used. The egg cells of many animals are large enough that DNA can be injected directly into the nucleus. Once it's there, enzymes that are responsible for DNA repair and recombination may help insert the foreign DNA into the chromosomes of the injected cell. Recently, scientists have started working with eliminating particular genes by constructing DNA molecules with two ends that will recombine with specific sequences in the host chromosome. Once they recombine, the host gene normally found may be lost or specifically replaced with a new gene. This kind of gene replacement can help pinpoint specific functions of genes in many organisms, including mice. And finally, cloning. A clone is a member of a population of genetically identical cells produced from a single cell. The technique uses a single cell from an adult organism to grow an entirely new individual that's identical to that original organism. Clones of animals were first produced in 1952 using tadpoles. The first mammal that was cloned was in 1997, a sheep called Dolly. Animal cloning is completed using nuclear transplantation. So what happens is we have an egg cell with a haploid nucleus. That's removed and replaced with a diploid donor nucleus from a body cell of the donor organism that produces an embryo. So like I said, first the nucleus of an unfertilized egg cell is removed. Then it's fused with a donor cell that contains a nucleus, an adult nucleus. That diploid egg develops into an embryo, which is implanted in the uterine wall of a foster mother. Since then, scientists have cloned cows, pigs, mice, and cats using similar techniques. Here's a diagram of what happened. So a donor cell is taken from a sheep's udder. This is a diploid nucleus. 2N. That's difficult to read. Hang on. 2N, donor nucleus. This haploid nucleus is removed. The two cells are fused using an electric shock, and then that fused cell begins dividing normally. Once it is enough cells that it's an embryo, it's implanted into a foster mother, and then this is how Dolly was produced. Now, Dolly didn't live a complete, full lifespan. She died early of cancer, I believe. So there's question to whether this is safe, whether it's something that is good to be used.